Hello everyone, welcome back to my channel Microbiology Easy Notes. If you like this channel, please like, subscribe, comment and share. Today's topic, Catalase Test. There are various ways by which microbes are identified in the microbiology lab. Biochemical testing is one of the methods to identify bacteria based on their unique metabolic and enzymatic activities. Among numerous biochemical tests, one of the tests is Catalase Test. Catalase is an enzyme produced by many aerobic and facultative anaerobic bacteria. Aerobic bacteria always need oxygen to survive, while facultative anaerobic bacteria can survive both in presence and absence of oxygen. Catalase was the first bacterial enzyme to be described by scientists. During cellular metabolic reactions, hydrogen peroxide generates as byproduct. Hydrogen peroxide is the reactive oxygen species. When accumulated in the cell, it causes serious effects on cellular functions and components which may lead to cell death. Catalase enzyme protects the bacteria from the toxic effects of hydrogen peroxide. It decomposes hydrogen peroxide into water and oxygen and helps the bacteria to neutralize this potentially damaging molecule. When oxygen releases from the cell, it escapes as bubble. Catalase reaction is the principle behind this test. During the observation, if oxygen bubbles or effervescence appears, it means bacteria present in the sample are catalase positive as catalase negative. Using this ability of catalase, we can easily differentiate and identify different bacterial genera and species. What is the purpose of catalase test? First is differentiation of gram-positive cocci. This test distinguishes between Staphylococcus species which are catalase positive and Streptococcus or Enterococcus species which are catalase negative. Next is identification of aerobic versus anaerobic bacteria. The test helps to differentiate between aerobic bacteria which produce catalase and obligate anaerobes which do not. So this test provides insights into bacterial respiration types. Differentiation among gram-negative rods. The Catalase test is commonly used to differentiate between various bacteria, and some gram-negative rod-shaped bacteria can be identified using this test. Specifically, many Enterobacteriaceae are catalase positive, while some like Campylobacter and Helicobacter are catalase negative. Identification of Mycobacterium tuberculosis. A semi-quantitative catalase test is used for the identification of Mycobacterium tuberculosis, aiding in its detection. Next is distinguishing Bacillus and Clostridium species. This test differentiates between Bacillus species and error-tolerant strains of Clostridium, which is catalase negative. It is crucial for accurate bacterial classification. What are the materials required for catalase test? The first is glass slide or petri dish or test tube, wooden applicator stick or sterile loop, 3% hydrogen peroxide solution, bacterial culture freshly grown, preferably 18 to 24 hours old, dropper or pipette. Procedure for catalase test. There are two types of method. The first is test tube method and the second is slide method. Slide method. First, heat sterile the inoculation loop. Cool it down. Take the sample from the test tube. Make a smear on the glass slide from the sample and move the slide over the heat 2-3 times. Cool it down. Keep the slide on the stand. Then add 2-3 drops of hydrogen peroxide solution to the smear. Observation. If bubble appears, it means sample contains catalase positive bacteria. If there are no bubbles, it means sample contains catalase negative bacteria. Test tube method. First, take 1 to 2 ml of hydrogen peroxide solution into a test tube. Heat sterile the inoculation loop, cool it down. Take the bacterial colony sample from the petri dish, then dip this inoculation loop into the test tube which is containing hydrogen peroxide solution. Observation. If there is no bubbles in the test tube, it means sample is catalase negative. If bubbles appear, it means the sample is catalase positive. Examples. Catalase positive bacteria are Staphylococcus aureus, Bacillus anthracis, Mycobacterium tuberculosis, Pseudomonas aeruginosa, and E. coli, etc. Catalase negative bacteria are Streptococcus pyogenes, Streptococcus pneumoniae, Lactobacillus acidophilus, Clostridium botulinum, Enterococcus faecalis, and Actinomyces viscus, etc.